The topic of today's lecture is Maintenance and Care of Laboratory Animals, Part 2. This lecture is in continuation of last lecture. We have already discussed these are five elements of main constituents of animal facility: physical, plant, environment, animal care, personnel, equipments, or standard operating procedures. We have already discussed this physical, plant, and environment in last lecture. We will take this animal care in this lecture and other these two components. Food is very essential element. One should check the feeding bowls randomly and observe whether the bowls have sufficient feed or not. One should check the quality of feed for fungal contamination. If any color change in feed, then it should be brought to the notice of concerned person. Check the type of diet and appropriateness of the feed for that particular species. Each species should be fed with a diet that is specific to them. The suitability to the species and suitability of feed depends on the composition of the feed. It differs from species to species. Appearance of the feed, presence of pesticides, presence of heavy metals. These are different types of pelleted feed for lab animals. Bedding. One should check for the cleanliness of the cages and inquire with the animal caretaker about the frequency of cleaning cages and the method and materials that are used for cleaning of cages. Animal bedding can influence experimental data and animal well-being. Bedding should be used in amounts sufficient to keep animals dry between case changes. Bedding should be appropriate for the species. Care should be taken to keep the bedding from coming in contact with the watering tube or leaks it, as the case might become flooded. These are different types of bedding materials. This is paddy husk and corn cob, wood shavings, paper cuttings or paper shreds. These are used as bedding materials for animals husbandry. A good husbandry program provides the environment, housing and care that permits animals to grow, mature, reproduce and maintain good health. Good husbandry minimizes variations that can modify an animal's response to experimentation. Elements of good husbandry include palatable, nutritionally adequate and non-contaminated food provided daily, water that is potable, bedding that is appropriate for the species, a sanitation schedule at the case, room and facility level that is conducive to maintaining the health of the animals, proper waste disposal, a sound program for pest control. These elements we will discuss in the coming slides one by one. Feed. Food made available to animals should be non-contaminated, palatable and nutritionally adequate. Areas in which diets are stored should be kept clean and enclosed to prevent entry of pests. Food should be stored off the floor and away from the walls on the pallets, racks or carts. Feed should be examined when received to ensure that feed order is correct and the feed bags have not been damaged or possibly contaminated. The date of manufacture or manufacturer's code and other factors that affect shelf life of food should be noted. Proper rotation of feed will assure that the oldest is used first and newest used last. In this example, feed is being stored inappropriately. The newer feed, dated 7th August, and has been placed on the top of the older feed, dated 28th July. Contaminants in food can have a significant effect on an animal's biochemical and physiological processes, even when present in concentrations too low to cause clinical signs of toxicity. Feed and chemicals should not be stored in the same room or area. Food should be stored and used in a prescribed manner, usually within 6 months after manufacture. Feed containing vitamin C usually has a shelf life of only 3 months and may benefit from being stored in a refrigerated environment. Purified and chemically defined synthetic diets are often less stable than natural ingredient diets. Their shelf life is usually less than 6 months, and these diets should be stored at 4 degrees centigrade or lower. This synthetic diet shown in the photo has become moldy due to improper storage. Feed containers used in animal rooms should be identified to preclude the use of outdated food. This feed container has a label that shows the type of feed, the date of manufacture and the date the feeder was filled. Feed containers should be cleaned and sanitized regularly. Feeders should be designed to allow for easy access to food with minimum contamination. In this example shown in the pic, an uncovered mouse feeder containing a specialty diet has been contaminated with feces and urine from the mice. Water. Randomly check the bottles for any leakage. If water leakage is there, then bedding material will be wet, which is uncomfortable to the animal. Animal should be given clean and ad libitum water in clean bottles. Animals should have access to potable, uncontaminated water. It is better to replace water bottles than refill them. If bottles are to be refilled, care should be taken to replace each bottle on the case from which it was removed. In this example, all the bottles have been removed from the cases simultaneously, and confusion as to where they belong might occur, leading to cross-contamination. Automatic watering systems should be checked at the case level daily to ensure proper maintenance, cleanliness and operation. It is also important to check the pressure reducing station. In this example, the water pressure of 9 psi is too high for rodents and they would not be able to drink from the liquid. Now we come to sanitation. One should randomly check the sanitary methods adopted by the caretaker and evaluate whether it is adequate. A clean atmosphere in animal facility should be maintained for the good health of animals, which involves bedding change as appropriate, cleaning and disinfection. The sanitation facilities should prevent cross-contamination. They should control aerosols, provides personal protection. The monitoring effectiveness of sanitation facilities is helpful in maintenance of health of animals. These facilities include use of vacuums, use of chemicals, etc. Sanitation involves bedding change, cleaning and disinfection of the housing unit in a manner conducive to maintaining the health of the animal. One should be aware of the cleaning schedule and how the housing units are marked. Red arrow to indicate date of sanitation or the date to be sanitized. Soil padding should be removed and replaced with fresh materials as often as necessary to keep the animals clean and dry. However, contact padding should not be changed in the animal room as shown in the right side of photo to prevent exposure of the staff to allergens and animal cages to possible cross-contamination. Primary enclosures can be disinfected with chemicals and or hot water. Temperature-sensitive tapes attached to cages or racks used daily will provide assurance that the cage wash unit is reaching recommended temperatures for adequate disinfection. In this photo, in one instance, the temperature was not reached. Animal rooms and support areas, that is secondary enclosure, should be cleaned regularly and disinfected as appropriate for the work conducted. Cleaning utensils should be assigned to specific areas and not transported between areas that pose different risks of infection. In the example at the right, the two hand brooms are from different rooms. Sanitation is to disinfect or to sanitize. There are 4 levels of sanitation: cleaning, sanitation, disinfection and sterilization. Cleaning means complete removal of visible soil from the surface. Sanitation is reducing the organism living on inanimate object to an acceptable public health standard. Disinfection is reducing the number of pathogenic organisms, not necessarily spores, to a harmless level. And sterilization is rendering an object free of all living organisms. And how to sanitize? The animal facility is continuously recontaminated by air, water, animals and people. The sanitation program includes cleaning and sanitizing. Degree of risk depends on the type and level of contamination and use. Greater risk of infection if organisms are resistant and or highly virulent. More risk if the animals are particularly susceptible, for example, immunocompromised animals. And how to choose a chemical? It depends on the following factors. The spectrum of activity is the first. The specific organism tested against the product. Effectiveness of the chemical in hard water. The hard water ions can inactivate the chemical. Third is the stability of the pH. Buffers prevent greater pH changes from the concentrated to the diluted form or by additives such as soaps. Use dilution judiciously. Using too much of product is wasteful and using too little may reduce or eliminate the antimicrobial effect. Contact time. It is essential that isn't be in contact with surface long enough to kill the most resistant organism present. Temperature. Heat could cause the evaporation of some of the components of the formulation. Then waste disposal. One should check the waste disposal mechanism thoroughly. Check whether biologic and hazardous waste materials removed and disposed of regularly and safely. Check whether adequate numbers of properly labeled waste bins are strategically placed. Conventional biologic and hazardous waste should be removed and disposed of regularly and safely in accordance with local and national standards. Waste containers should be leak-proof and equipped with tight-fitting lids. In the example at the right, the cover is missing in the biohazard container. Emergency, weekend and holiday care is very important. Check whether proper arrangements are made for maintenance of animals on weekends and holidays. One should check the arrangements made in case of emergency and also disaster management at the animal facility. In the event of an emergency, institutional security personnel should be able to reach people responsible for the animals. Pest control, check whether proper pest control, insect or wild rodent measures are followed at the facility. And transport of laboratory animals. One should check whether the transport of animals from one place to another is undertaken with care. Check the mode of transport for animals. It depends on the distance, seasonal and climatic conditions and the species of the animals. Check whether the containers are of appropriate size and comfortable for free movement and protection from possible injuries. Quarantine and stabilization are also very important. The animals must be quarantined before inducting into the main colony. Duration of quarantine depends on the species procured. Period of quarantine for various species is as follows. For small laboratory animals, it is 7 days. Genetically modified lab animals, it is 14 days. Dogs for research purpose and monkeys and large animals, it is 6 weeks. Veterinary care of animals. Proper veterinary care should be provided to the animals. If an animal is any animal is sick, report to the veterinarian for further action. Animals should be cared for by qualified personnel every day, including weekends and holidays. In the event of an emergency, personnel should be able to reach individuals responsible for the animals. In the example at the right, personnel on call are posted on a bulletin board in the animal facility. The duty chart contains the date, veterinarian's name, contact number, supervisor's name, contact number. Now we come to the identification and records, which are very important. Methods of animal identification include room, track, pen, stall and case cards with written or barcoded information, collars, bands, plates, tabs, colored scales, ear notches and tags, tattoos. Subcutaneous transporters are acceptable methods of identifying animals. Clinical records for individual animals are valuable sources of information, particularly for dogs, cats, non-human primates and farm animals. The record should include pertinent clinical and diagnostic information, date of inoculations, history of surgery, post-operative care, as well as information on experimental use. The identification card should include the names and means of contact of the responsible investigators, protocol number, species, strain, and other pertinent information about the animals. The case card at the right is a suitable example of a properly prepared rodent case card. Contains investigator's name, protocol number, induction date, whether it is male or female, age, weight. In this example, a case card has been changed. The question should be who changed the card, why, and are the animals being used appropriately in an approved protocol of similar nature and or pain or distress category. Adequate veterinary care consists of effective programs for preventive medicine, surveillance, diagnosis, treatment and control of disease, including zoonosis control, anesthesia and analgesia, surgery and post-surgical care, assessment of animal well-being, euthanasia. We will discuss these in the coming slides. Then procurement for transportation. The animal should be acquired lawfully and the receiving institution should attempt to ensure all transactions are conducted in a lawful manner. The method of transportation should be appropriate for the species to protect the animals from injury, exposure to the elements, or possible contamination. Preventive medicine. The effective preventive medicine programs enhance the research value of animals by maintaining healthy animals while minimizing possible variations associated with disease and in apparent infection. These programs consist of various combinations for policies, procedures and practices related to the quarantine and stabilization of animals by species, source and health status. Then health monitoring is very important. It is done to answer the following questions. Are the animals in the unit clean? If not, what are they contaminated with? Health monitoring program mainly includes screening of animals for viruses, bacteria, fungi and parasites. The design of health monitoring program depends on housing condition, research activities, frequency of introducing animals, likelihood of interference, type of experiment, the implications of infectious agents on experiments. Rodent pathogens are not only hazardous to animals but also to humans and severely influence the animal experiments. Some are pathogenic and may induce clinical signs with variable morbidity and mortality. Most of the microorganisms induce mild disease and silent infections are often activated by experimental procedures like stress, toxic substances, tumors, or environmental influences such as transportation, suboptimal humidity and temperature. The design of monitoring program we have already seen in the previous slide. It depends on research activities, housing condition, whether the housing is conventional or barrier-based, type of experiment, long-term or short-term, frequency of introducing animals, likelihood of interference with the research, and methods of sampling may be of two types: random sampling from all areas or sampling from different age group of animals. The frequency should be once in 3 months and size at least 10 animals at a time. Collection of material for viral samples. Serum is collected for bacterial, fungal, nasal, tracheal, lungs, pupils, vagina, cecum may be collected. For parasitological examination, skin or hair for ectoparasites, fecal contents, serum for serological testing. Then how to control the disease. All animals should be observed for signs of illness, injury or abnormal behavior by personnel trained to recognize such signs. This should be done at least daily but more frequently if the situation warrants. Professional judgment should be used to ensure the frequency and character of the observations minimize risk of the involved animals. It is imperative that appropriate methods exist for disease surveillance and diagnosis. Death, signs of illness, distress, and other deviations from normal should be reported promptly to ensure appropriate diagnosis and treatment. Prevention of disease. It is often thought of disease prevention is often thought of as simply buying animals with a known health status. But what about the animals that have been in the colony for a long time? In the example at the right, it appears that a preventive dentistry program is needed to assure this dog has a clean and healthy oral cavity. Reporting of disease. The use of an animal observation and reporting card, similar to that shown on the right, is an effective way to ensure timely animal observation and reporting of deviations in the animal's condition. This copy is for veterinarian. An identical copy of this card is placed on the case of the affected animal, so the animal is easy to locate. In this, the investigator's name is present, species, strain, date, project, ID number, date of birth, line, sex, room, case, location, and technician's signature is here. The back of the animal observation card has spaces for the technician to indicate the sign or of the symptom observed. Identical to the back of the card given to the veterinarian, this card alerts all technicians to the animal's condition or situation. Treatment of disease is very important. Color coding of drugs for expiration date is only one effective manner to assure that effective medical care is available within the animal facility. The presence and possible use of outdated drugs is a deficiency and a suitable program should be in place to prevent their use. For surgery, appropriate attention to pre-surgical planning, personal training, surgical and aseptic technique, and animal well-being during all phases of surgical protocol will enhance the outcome of the surgery. Pre-surgical planning should include input from all members of the surgical team. It is also important that all personnel have appropriate training to ensure good surgical technique is practiced. One easily overlooked aspect of pre-surgical planning is the storage and use of surgical instrument sets. The example on the right shows a surgical bag that has been sterilized and plated. check for the sterilization date and the sterility expiration date. Survival surgical procedures may be classified as major or minor. However, both should be completed using a septic technique. Although major surgery usually envisages the use of a large surgery suite, rodents also may undergo major surgery. In the example at the right, proper procedure and practices are being followed. Marking a case card to indicate the animals have undergone surgery is a convenient way to alert the care staff of the special needs or condition of the animals. Socialization of animals is very important. The social environment usually involves physical contact and communication among members of the same species and the caregiver. When appropriate and compatible with the protocol, animals should be housed in physical contact with conspecifics, same species animals. Environmental enrichment is also a very important element. The foraging board is attached to a non-human primate case. While animals in the wild invest substantial time in foraging for searching food, animals in captivity are usually deprived of foraging opportunities. However, they will readily work for food even in the presence of freely available identical food. What other enrichment devices are used? Is there a schedule for rotation? We will see in coming slides. There are mainly two types of environmental enrichment: social enrichment and physical enrichment. Social enrichment means socialization of animals with conspecifics and or contraspecifics, including humans. A social partner leads to an increase of alertness and exploratory behavior and provides diversion, occupation, and also some feelings of security in stable, harmonious groups. The physical enrichment is again divided into three types: complex enclosures, sensory stimuli, nutritional stimuli. The first type is complexity. Means appropriate structuring of the cage or pen environment is more beneficial than provision of larger floor area. Most rodents and rabbits attempt to divide their living space into separate areas for feeding, resting and excretion. Such divisions may be facilitated by structures within the cage, for example shelters, nest boxes, nesting materials, tubes and platforms that provide withdrawal areas and lookout possibilities. The provision of nesting materials, it is seen that it enhances the breeding results in mice and rats. Rats, mice, hamsters and gerbils, the nesting material is provided for resting and breeding purpose. And it is important to provide nest boxes or other refuges for rats. For guinea pigs, refuges such as nest boxes, tubes or shelters are provided. Wood sticks for chewing and gnawing, soft wood sticks are provided. They are having a habit of chewing the objects. Gerbils, for this the thick layer of litter approximately 20 cm long are provided for digging and nesting behavior. Nesting material, for example hay or straw are provided, and wood sticks for chewing and gnawing are provided. Hamsters, for hamsters nesting material, refuse area, tube or hut, roughes and the gnawing objects are provided. For rabbits, minimum roughes, hay blocks or chew sticks, and as well as an area for lookout and withdrawal possibilities, like for example platform is provided for them. For breeding doors, nesting material, nest box or other refuse are provided. Toys can have a beneficial effect on the animals in development of their exploratory behavior and in locomotor and visual development of their visual performance. Second is sensory enrichment. Most satisfying enrichment for rodents and rabbits is visual, auditory, olfactory and tactile communication with conspecifics or contraspecifics, either directly or through bars. Cage cleaning is necessary routine procedure in lab animal facilities. Providing different food items, for example carrots for rabbits, seeds for rodents, may act as a taste stimuli. Third is nutritional enrichment. Presentation of food and giving the animal the opportunity to forage, for example scattering food in the bagging, appear to prevent boredom because in nature a large part of time budget is spent on this activity. Even though these food items might be contaminated by feces and urine, when the animals have no opportunity to compartmentalize their environment. The frequency and schedule have an impact on animal. Feeding rabbits immediately before dark in their active period instead of in the morning reduced stereotypic behavior remarkably. For guinea pigs and rabbits, additional food items, hay, straw, or grass cubes can satisfy the need for roughaging and for chewing. Wood sticks, aspen blocks are provided to them. Now, the fourth constituent of animal facility is personnel. Appropriate personal protective equipment should be available at the door of the animal facility. It will protect from the possible extraneous contamination. Lab coat, shoe covers, and safety glasses are among the more popular PPE. For personnel and training, check whether personnel caring for animals are appropriately trained. If not, formal or on-the-job training should be provided to facilitate effective implementation of humane care and use of animals. Check whether personnel are properly trained regarding zoonosis, chemical safety, microbiology, physical hazards, and handling of waste material. Check whether personnel use protective clothes while entering or handling of animals. Check whether periodic medical evaluation of animal caring staff is done. Fifth and last is equipments or standard operating procedures. The standard operating procedures related to animal facility activities. Equipment in the animal facility must be available at the workplace. Record keeping is very important. Every facility should maintain proper records and should be made available during inspection. Some of the important documents to be checked are as follows. Form C is the number of animals, record for the number of animals that is acquired or bred, and it is maintained on daily basis. Form D is the record for the number of animals acquired by the investigator, and in this the IAC protocol approval details are present. Other important records are animal house plans, which includes typical floor plan, all fixtures, etc. Animal house staff record, both technical and non-technical, health record of the staff or animals, all SOPs relevant to the animals, breeding, stock, purchase and sales record, minutes of institutional animal ethics committee meetings, death record, clinical record of sick animals, training record of staff involved in animal activities, and order and basis report. Then equipment, appropriate and adequate equipment should be available. For equipments, the strict program of validation, qualification, calibration and maintenance is very critical. Appropriate logbook should be maintained. The problems associated with managing the animal facilities include procurement of good diet, that is standard feed, management with the limited budgets and resources, handling of supporting staff, their recruitment, training, incentives, leave habits, procurement of bedding materials, matters related to ethical committee. These are the references for making this presentation. This is all about maintenance and care of laboratory animals.